my name's Adeline McKie. I'm doing a PhD at Queen's University Belfast, and today I'm going to present my project, which is investigating cancer vulnerabilities for drug discovery. Synthetic lethality is a well-known genetic dependency. For example, as shown in the image, there is a synthetic lethal relationship between BRCA and PARP genes. In healthy cells, PARP inhibition is not lethal, and the BRCA2 mutation is common in cancer cells and does not cause cell death. However, PARP inhibition will cause selective killing in BRCA mutant cells, and this provides opportunities for precision oncology. Multiple myeloma is an important application area in my PhD. This is the second most common blood cancer and arises from plasma cells. The current 10-year survival for multiple myeloma is only 29%, and despite the advancements in therapeutics, almost all patients will develop relapse and resistance. Therefore, there's a need to identify more effective therapy. My PhD seeks to identify more effective therapies in cancer and to build upon computational approaches developed in the Overton group, such as SynLeg, which is a web resource to predict candidate gene dependency relationships. And SynLeg uses the MultiSEp algorithm for the discovery of candidate gene dependency relationships in multiomics data. And the results of benchmarking MultiSEp were published in the SynLeg paper before I started my PhD. And the MultiSEp algorithm was compared against DAISY, which is a gold standard method in the literature for predicting synthetic lethality, and BiSEp, which was the precursor to MultiSEp, and it looks at two expression modes to predict synthetic lethality. And this was using gold standard data from SynLeafDB, which is a comprehensive database of curated synthetic lethal interactions with a confidence score. And importantly, from these graphs, it shows that MultiSEp outperformed both DAISY and BiSEp. MultiSEp had a lower false discovery rates, a larger area under the curve, and higher coverage for predicting synthetic lethal relationships from SynLeafDB. And I've been analysing transcriptome data to predict gene dependency relationships by the application of regularised Gaussian mixture modelling, and this is to determine gene expression clusters. And the figure here highlights how I use this method to cluster samples according to the expression of two genes. And the arrow in the figure highlights for the expression of two genes, there is a depletion of samples in the bottom left cell of the contingency table. And this is where both genes have a low expression, and this is characteristic of synthetic lethality. So I've developed a statistical approach to detect depletion of samples in the bottom left cell using the binomial test and the Poisson distribution. And I've incorporated this function in the development of the MultiSEp R package, and I've added 63 unit tests, some new functionality and documentation, and the MultiSEp R package is planned to be released this summer. And we've developed a high-performance computing enabled workflow for the discovery of multiple myeloma gene dependency relationships at genome scale. And we've used the COMPASS study, which is a rich resource of multiomics data for multiple myeloma. And I've taken the transcriptomic data to predict gene dependency relationships for 27,288 genes with over 372 million candidate interactions. We then applied two Q-value thresholds, firstly for gene dependency relationship significance, and secondly to identify synthetic lethal depletion patterns. We then integrated the mutational data for the COMPASS cohort, and we used the Varity, SNP-EFF and SNP-SIFT algorithms to predict deleterious mutations, and this was in order to assess the therapeutic coverage across myeloma patients. And this slide shows the visualization of the predicted synthetic lethal network for multiple myeloma. And the node colour represents the predicted population coverage assessed by the frequency of mutations in neighbours of the candidate gene. And 10 hub genes were identified, and these are annotated on the network as stars. And a drug targeting any of these star genes is predicted to have a therapeutic benefit for at least 5% of myeloma patients. And importantly, from this network, many genes are involved with the ubiquitin proteasome system, and this is dysregulated in multiple myeloma and a target of current frontline therapy. And 2 of these hub genes within the network achieved 58% coverage, which means 58% of patients would benefit from a drug targeting these hub genes. However, targeting one of these genes may cause side effects where bone density is reduced, and this is an important health issue in multiple myeloma. Therefore, this gene was not prioritised. And to validate our predicted synthetic lethal network for multiple myeloma, we use CRISPR screen and gene expression data from the cancer dependency map. We also validated predictions by performing chemical sensitivity analysis with datasets from the Cancer Cell Encyclopedia and the Cancer Therapeutics Response portal. And the validated synthetic lethal network is shown on the right, and this consists of 2,739 genes and 4,390 validated edges. And the node colour within this network represents the type of validation and if a gene was found in more than one validation dataset. We then measured the dependency profiles of genes present in the synthetic lethal network, and we found 56 genes had significant myeloma-specific dependency profiles. And the box plot shows an example of the CRISPR dependency scores for CCND2 across different cell lines. And the plot shows that the loss of CCND2 has a selective dependency in myeloma cell lines as shown by the negative CRISPR scores that correspond to a loss of cell viability. So in summary, we have developed the MultiSEp R package to discover mutually exclusive loss signatures in multi-omics data, and we've applied the MultiSEp R package to generate a high-confidence synthetic lethal network. And we've used this network to predict candidate drug targets with efficacy for many patients. And we validated our findings of the synthetic lethal network. And I'd like to say thank you to my supervisors, Ian, Mark and Hans, for all their support and guidance throughout the project, and to the Overton group for all their valuable guidance. I'd also like to say thanks to Lisa for helpful discussions around multiple myeloma, and to Fritz and Joe for their helpful insights around the Varity threshold values. And thank you for listening. I'm very happy to take any questions.